Should we fly into space to avoid contact with antigens? No. Hi, I am Sahili Chatterjee and welcome to my second lecture video on antigen processing and presentation. So, let's try to know what are these antigens. Antigens are molecules that are recognized by an organism as foreign and is capable of evoking immune response inside the host body, either alone or by forming a complex with another molecule. Every antigen possesses a distinct property called antigenicity, which is the ability of the molecule to either combine with an antibody or any other cell surface receptor. Antigens that are capable of inducing adaptive immunity are widely called as immunogens. To counter the impact of these antigens, various immune-responsive cells comes in the area of infection. The first one is the B cell that arises and matures inside the bone marrow. B cells have receptors called antibody on their surfaces that binds with the antigen present outside the cell, thus neutralizing its effect. The second one are the T cells arising in the bone marrow but travels to the thymus to mature and develop. The T cells fight with antigen present inside the cell through a process called antigen presentation, wherein an antigen is presented to them by various antigen presenting cells by expressing MHC molecules. So, let's now discuss about the MHC molecules. MHC or major histocompatibility complex molecules are glycoproteins that plays an integral role in self and non-self discrimination and development of both humoral and cell-mediated immunity. It is having major two variants. Let's discuss the first one. Class 1 MHC molecule. Class 1 MHC molecules are an assembly of 3 alpha chains that associates with a beta-2 microglobulin protein and presents antigen to cytotoxic T cell. Class 2 MHC molecule. Class 2 MHC molecule is a heterodimer of 2 alpha chains and 2 beta chains that presents antigen to helper T cells. Pathways. Presentation and processing of the antigens by the MHC molecules proceeds in two distinct ways, precisely classified as exogenous pathway mediated by class 2 MHC and endogenous pathway mediated by class 1 MHC. Now, let's see what cellular activities takes place in displaying an antigen by the exogenous pathway. In the first step, antigen-presenting cells phagocytose antigen either by simple endocytosis or receptor-mediated endocytosis, forming an extracellular vesicle inside the cell. In the next step, these extracellular vesicles matures to form phagolysosome, within which the antigenic peptide is degraded into oligopeptides due to the action of several hydrolytic enzymes and varying pH. During this time, the assembly of the class 2 MHC protein concludes in the rough endoplasmic reticulum, and the protein associates with an invariant chain to prevent endogenously derived peptides from binding to it. The invariant chain also assists in the exit of the glycoprotein from the rough endoplasmic reticulum, its routing via the trans-Golgi network, and subsequently dispatched to the endocytic processing pathway. The invariant chain gets degraded while moving from early to late endosomes. However, a short fragment termed as CLIP or class 2 associated invariant chain peptide remains bound to the molecule, preventing premature binding of antigenic peptide. The final antigenic protein degradation and peptide loading occurs within the MHC class II containing compartment, a unique form of endosome found only in antigen-presenting cells. Eventually, CLIP gets exchanged with the antigenic peptides in presence of HLA-DM. HLA-DO inhibits the functioning of HLA-DM, thus preventing the dissociation of CLIP. After the peptide is bound, the peptide MHC complex is transported to plasma membrane where the complex acquires a stable and compact form. We'll now proceed to understand the endogenous pathway by which cytotoxic T cells are presented with antigen. Inside an eukaryotic cell, proteins are carefully regulated and are degraded to their constituent amino acids and recycled. However, some persist in the cytosol as peptides. These peptides bind with ubiquitin and the conjugate complex so formed then pass through the proteolytic system, the proteasome, which consists of a 19S regulatory component and a 20S base. The antigen is degraded into small peptides inside the proteasome. The peptides produced in the cytosol pass through TAP by an ATP-driven process. The assembly of these peptides with class I MHC molecules involves processing by a group of chaperone proteins, namely calnexin and ERP57. These chaperones together bind with the alpha component of the MHC. Later, beta component binds with the alpha component by removal of calnexin. Eventually, other chaperone proteins like calreticulin and tapasin binds. Another non-MHC chaperone, ERAP1, processes the peptide by removing the amino terminal residues such that the peptide fits into the peptide groove of MHC complex, and in between, three chaperones, calreticulin, ERAP1 and tapasin, dissociates from the complex, allowing the peptide conjugated MHC to exit the rough endoplasmic reticulum. Finally, the class I MHC molecule loaded with the peptide is transported to the cell surface for display of antigen to the T cells. Thanks for watching this video. Please like, subscribe and share.
This video is brought to you by Science and Technology in association with Banerjee Infotech Solutions. For more information and assistance, kindly contact on the following mobile number given below.